how the foundation is going to expand out beyond exfoliation syndrome. I must say that Elena has really energized the board in terms of thinking about these things, and I think that is what brings everyone to the table, and it's exciting to see the board energized in and around this topic. Just need to figure out how to advance. Okay, so these are my financial disclosures. I actually do receive NIH grant support to study primary open-angle glaucoma and exfoliation syndrome, and I'm very fortunate to do so, and really kind of helps to influence my thinking about these topics. But Elena asked me to sort of start out with basics, and that is where I will begin. And this is what when I see patients who are newly diagnosed with glaucoma, they often ask, what is glaucoma? And I explain that glaucoma is a disease of the optic nerve, and you see the optic nerve here, and that the optic nerve is like a telephone cable that connects the eye to the brain. And then in glaucoma, there's a premature deterioration of those fibers. We're born with roughly 1 million fibers, and in glaucoma, those fibers deteriorate. The number one reason why they deteriorate is because of elevated pressure inside the eye. Here is a picture of a normal optic nerve, and you can sort of liken it to a donut with a pink rim. And then in glaucoma, this is an advanced glaucoma case, that pink tissue disappears. The reason why that happens is because the fibers in that optic nerve cable have deteriorated. And typically this occurs because of elevated pressure inside the eye. Now, there are many different kinds of glaucoma, and one way to start to think about them is to classify them into open angle glaucoma and closed angle glaucoma. And just to try to give you an analogy of what's going on, I like to use, in terms of understanding the difference between angle closure glaucoma and open angle glaucoma, I use what's called a kitchen sink analogy. So here's a picture of the front of the eye, and I'm pointing to the faucet, so to speak. This is where the fluid is generated. So in the eye, the faucet is always on, and the drain is supposed to be always open. But in angle closure glaucoma, something happens where the peripheral iris tissue acts like a stopper to close the drain. Now, the eye is a little bit stupid in that it just keeps generating fluid at the same rate. Now, in your kitchen, what happens is the water, if that happens, if the drain gets closed off and you keep the faucet on, the water is going to build up and fall on the floor. Inside the eye, the water can build up and rapidly damage those fibers in the optic nerve, leading to considerable vision loss. Now, we also have open angle glaucoma. And in this situation, the faucet's always on. We look at the drain and it looks like it's open, but the pressure can still build up inside the eye. And one of the analogies that I like to use is that there can be little pieces of lettuce stuck deep down in the drain, and that is what's causing the fluid to build up in the eye. Turns out that that's a decent analogy for exfoliation glaucoma, because in exfoliation glaucoma, little fragments of exfoliation material and pigment lodge deep into the drain that cause the pressure to elevate. Now, the way I like to look at a disease that's not completely understood is to develop a timeline. So you start out in exfoliation syndrome with normal ocular structures. Then there's what we would call a preclinical phase. Something happens. We don't know what that something is, but then that something leads to the accumulation of the exfoliation material in the front of the eye. That material clogs the drain, causes the pressure to go up, we get optic nerve damage, and other problems that can develop in exfoliation syndrome. And ultimately, you get visual disability. So this is the timeline. And exfoliation syndrome is a wonderful disease to study because we have this biomarker. This biomarker is this whitish material on the surface of the lens here that's accumulating. And so that's what makes this an interesting disease to study. And if I were to define exfoliation syndrome, and this is a more magnified view of the front of the eye, the pupil is dilated, and this stuff here is the exfoliation material, it's that it's an extracellular deposit disorder. That is, deposits occur outside of cells. And so if we take a histology slide of the lens and we look at it, this is the surface of the lens, and this stuff right here is the exfoliation material. And notice that there's a little pigment granule in there as well, because pigment gets stuck in the exfoliation material. So this is the biomarker for the disease, and this is what we've been trying to understand, why this stuff accumulates in the front of the eye. Now, it's really interesting. I think there are only 3 diseases, and maybe Dr. Kaufman could correct me, maybe he knows other diseases. There's only 3 diseases that are categorized by extracellular deposits. Exfoliation syndrome, of course, age-related macular degeneration, where you get this drusen-like material that accumulates outside of cells. This is a picture of the retina. And Alzheimer's disease, which is a disease of the brain, where you get these neurofibrillary tangles that occur in the extracellular spaces of the brain. So if we're going to understand this disease, I think that we really need to sort of address certain elephants in the room. When I was a resident, I read in my books that Scandinavian ancestry was a risk factor for exfoliation syndrome. And that was because 1 in 5 people over the age of 60 living in places like Sweden and Iceland, etc., Finland, that 1 in 5 over the age of 60 developed exfoliation syndrome. However, that doesn't mean that your ancestral genes for being Scandinavian is what's contributing to the disease, because there's no one else to compare these patients to. So we did a study in the United States where we have a more heterogeneous population of folks, and we asked the question, is Scandinavian heritage a risk factor for exfoliation syndrome? And the answer that we found was no. So there's something about living in those countries that contributes to the disease, but it's not necessarily their genetic makeup. So that's an interesting observation. Another important elephant in the room is the question about whether this condition is rare in African people. Certainly, if we man ophthalmology clinics here in the United States, we find very quickly that African ancestry is a risk factor for primary open-angle glaucoma. But we rarely see patients with exfoliation syndrome of African ancestry in the United States. However, in a paper review, we scoured the world's literature with Dr. Rich, and we found out that if you look in Africa, there are places where you find absolutely no exfoliation syndrome. And this was a really good study in Tanzania. They looked at 4,000 people. They found no exfoliation syndrome. However, go right over to Ethiopia, go over to Ethiopia, and there's a high clinical burden of the disease. Furthermore, in South Africa, the prevalence of exfoliation syndrome among people of African descent is surprisingly high. So again, there's something about different locations that are contributing to the disease. And I think it's environmental, but we can talk about that at the end of the talk. But really, so what causes the exfoliation material to form in the eye? After all, this is a critical question because it is this material that lodges in the trabecular meshwork or the drainage angle of the eye to lead to elevated intraocular pressure. This is the little pieces of lettuce that are stuck deep in the drain that cause the pressure to go high. And so you could look at the histology of this, and this is the anterior chamber, this is the trabecular meshwork. This is where the fluid inside the eye is trying to go out the drain, down the pipe that we call Schlemm's canal. And this is the material, the arrows are pointing to the material that are clogging the drain. And it can get pretty severe. You can see this material is almost completely closing off the deep aspect of the drainage system of the eye. And that's why these patients can present with very high intraocular pressure and a lot of glaucoma damage. So I hope that I've piqued your interest into this question. And so one basic question is, what's the distribution of this material in the eye? And the answer is, we looked at this very carefully. There are lots of studies that looked at this. And for the most part, most of this material is in the front of the eye. This is a post-mortem specimen of an eye, and the backside of the eye has been removed. You're looking at the lens from the backside. It's cataracterous. And these are what are called the ciliary processes. They make the fluid inside the eye. And you could see that there's a lot of exfoliation material on the ciliary processes. It's coating the zonular fibers. This is what connects the eye to the lens to the eye wall. And it's coated with exfoliation material. But once you get beyond here and you start looking in the retina, no material. So most of this material is in the front of the eye. Now, another question is, what is this stuff made of? What is the components of the exfoliation material? After all, if the strategy is to find out why the material forms in the first place, or if we're going to develop strategies to disassemble the material so as to make the drainage system of the eye work better, we need to know what the material is. There have been lots of studies on that, and I'm not going to bore you with the names of the stuff because your eyes are going to glaze over, at least most of your eyes will glaze over, but leave it to say that it's many different large molecules, most of which are normally part of a cell's basement membrane. What is a basement membrane? Well, I like to, again, another analogy, remember I used the kitchen sink analogy, this is the topsoil analogy. So cells, surface cells inside any structure in the human body sit on a foundation that we call a basement membrane. Let's liken it to your backyard. You have a lawn, you have the grass, and the grass is stabilized by topsoil. So we can think of this basement membrane as like a topsoil that helps to stabilize surface cells. Now, inside the eye, it's unusual in that we have basement membranes that face fluid-filled cavities. So in other words, it's like inside the eye, the motif is the topsoil is up and the grass is down, and that the basement membrane is facing fluid-filled cavities, which might be why the exfoliation material tends to accumulate very prominently in the front of the eye. So again, now let's just bring this back to the eye, and this is a histological section. In other words, looking at eye tissue under a microscope under high power, and we're looking at the lens. And so this is the surface of the lens. This is the basement membrane or the topsoil. These are the epithelial cells. This is a fluid-filled cavity. This would be the anterior chamber of the eye, and this is the exfoliation material. So again, taking my upside down lawn analogy and putting that right next to the human lens, you can see the topsoil is like this pink basement membrane. The cells are underneath. And then what happens in exfoliation syndrome is something goes wrong here in the topsoil and something starts spouting out that's abnormal. If we go and look at even higher magnification, let's say we're going to take an electron microscope and we're going to really focus down on one cell. What would it look like? This is the surface of one cell, and they have these curious V-shaped invaginations, and spouting out is this electron-dense material that seems to go all the way through to the capsule and come out onto the surface. And this is the exfoliation material under high magnification. So what we think is going on in exfoliation syndrome is these cells, these epithelial cells are under stress, and they do something that causes this accumulation of exfoliation material on the surface of the anterior segment structures of the eye. So my one hypothesis that I have been exploring is that one of these stressors is ocular UV exposure. So the hypothesis that I have is that exfoliation syndrome is a form of the deleterious ocular aging, whereby lifelong ocular UV exposure somehow triggers dysregulated basement membrane metabolism. It disrupts that topsoil and causes this stuff to spout out from the surface. So let's bring this back to clinical reality. I think with COVID, we have not been able to travel anywhere. But this is a picture of Greece, the Santorini island. And notice that you've got nice blue water. You've got a lot of buildings with white backgrounds. This is a perfect place for sun to bounce off the water or off the structures and into the eye, and over long periods of time contribute to the development of exfoliation syndrome. It turns out that the prevalence of exfoliation syndrome in Greece is about 10%. And a lot of clinical studies about exfoliation syndrome come out of Greece because the condition is so common there. So if we put this in a schematic, now we don't stare at the sun all day. And so what I think is going on is when the sun is rising or setting, the sun rays are weak and they come into the eye, but there's not damaging radiation. But when the sun gets high in the sky, we're not staring at it, but that sun that bounces off that blue water or those white backgrounds into the eye over long periods of time could be contributing to exfoliation syndrome. So now if we go back to our detective diagram, what I'm saying is one of the factors, and I'm not saying this explains all of exfoliation syndrome. Believe me, there's probably many other things going on, and there are probably plenty of people who've not been exposed to enough UV radiation to get this condition. But that one of the things that could be happening is long-term ocular UV exposure is part of the preclinical phase of the disease. And so that, if that's true, that would have important public health implications, because it would suggest that the disease really starts during young adulthood. Exfoliation syndrome is largely regarded as the disease of someone who's in their 60s and 70s, and that it's really rare in their 20s, 30s, and 40s. But I think that this disease is actually brewing over a very long period of time. And it might also...
My apologies. Sal raised his hand. I think he has a question.
Okay. I don't see the question, so you're going to have to stop me if you've got them. I'm happy to take it.
So on what's on the screen right now, and given what you just stated about it being a long-term accumulation, that makes sense, a longitudinal issue. Has there been studies in that same location in Greece for outdoor workers versus indoor workers, farmers versus office workers? And you would think the prevalence would be higher in the farm workers than in the office workers. Is that something that's been toyed around with as a thought?
Sal, excellent question. I'm glad that you asked it. So there was a study on the island of Rab. I don't know if anybody knows where the island of Rab is, but it's a little island in the Adriatic Sea, pretty close to Greece. And they did a study, and they looked at people who were working like fishermen and outdoor people compared to people that had indoor jobs. And guess what? All the exfoliation was in the people who worked outdoors, fishermen, etc. And almost none in the people who worked indoors for the most part. So yes, there's a lot of construct validity for this idea. And I can go on and on and on about that. But you know, the long and the short of it is that studies that we've done in the United States, studies that have been done in India, indicate that more time spent outdoors in general is a risk factor for the development of exfoliation syndrome. Any other questions that anybody wants to raise at this point? If not, I will. Did that answer your question, Sal?
Yes, it seemed obvious to me that that would be an avenue of investigation. And, you know, the only thing outside that's not inside of consequence is the sun. So it tends to speak to your issue, right?
Right.
I can't think of any differentiator between daylight time and sunset and who's inside and out differentially that way from a work point of view, but that would be another area that'd be sort of interesting to see it's the high noon kind of stuff that's causing us the problem.
Right, right. And so if we get back, what I'm showing now is, imagine if there was a lot of sun around, but you live in an area where there isn't a lot of reflected light, like a very lush green environment with a lot of grass. Grass doesn't reflect light very well, so there's less reflected light in the eye. And so now if we go back to our diagram about Africa, we might understand why Tanzania, which is a lush green area, has no exfoliation syndrome, and Ethiopia might have a lot. Now, Ethiopia, these are two countries, Tanzania and Ethiopia, they're fairly close to the equator, but Ethiopia is a very mountainous climate, and so it has a more temperate climate, and there's less grass and more mountainous types of structures, more sand, and in fact, once you get into Saudi Arabia and places like that, it's all desert. And so these environmental factors allow for more sun to bounce off the ground into the eye. And so just to follow up, Sal, another thing that we've done is, because it's so hard to find a metric that measures what your ocular UV exposure is through your entire life, we asked the following question: does a history of a basal cell carcinoma or a squamous cell carcinoma, these are skin cancers, largely of the head and neck, there's pretty strong consensus that UV exposure during the course of your life is a risk factor for developing these skin cancers. Are these types of cancers associated with increased risk of exfoliation glaucoma? And the answer is yes. There was about a 40% increased risk of subsequent development of exfoliation glaucoma if you had a history of these skin cancers. And this is a paper that Dr. Rich and I and my other colleagues have published in the Journal of Glaucoma.
So Louis, let me ask you a very quick question on that, because it occurs to me that did you limit the skin cancer to forehead, cheek, and nose, as opposed to back and elbow or whatever, you know, because your arms are short sleeves. So it's facial skin cancers, because that would probably correlate closer to the eye, and then maybe that 40% would be 80%.
Right. Well, great question again. And because this is a nationwide database, we weren't able to get that localization. The majority of them were head and neck, but we can't say that for sure. So we're doing a follow-up study at New York Eye and Ear where we can ask that kind of question. And you know, for those people who tell us that they have these cancers, we could do more of an investigation of where those skin cancers occurred. And in fact, what we want to do in follow-up is to say, do these skin cancers happen more on the side of the eye with the worst exfoliation glaucoma, right? Because that adds even more fuel to the fire. So that's what we're doing right now. We've recruited about 190 patients, but we need to do at least another 100 more before we can answer that question. But yeah, we're following up on that.
One more question, and then I'll stop with questions.
No, that's great. These are great questions.
If we work in an environment where radioactive material or even just X-rays, we have these badges that we wear, right, to collect the amounts of rads that you accumulate.
Right.
So I wonder if it makes any kind of wild sense. It occurred to me that if you wear glasses, you're already wearing an artificial device on your face. So if you wear glasses and you could have a UV cumulative count from wearing glasses and have a longitudinal study, in 10 years, you could have the amount of UV in my eye differentially from yours, because I see you wear glasses. I assume you wear them all the time. You forget that if it's reading or whatever. If a small enough UV calculator could be on a pair of glasses, in 10 years, we could get like super hyper calculable and plottable data on UV exposure. Does that make any sense, and is that possible?
It makes sense, but it's hard to do. That thought experiment is a wonderful thought experiment, Sal. And I've thought of that experiment too. And it's hard to come up with that metric. That's why we did this study about the cancers and their relationship to exfoliation glaucoma, because it's the next best thing, right? So to do the study you described, we'd have to get a lot of people wearing UV counters, so to speak, glasses for long periods of time. And also keep in mind that like the cornea will not allow in very short wavelengths of light. So the cornea lets in actually very small amounts of UVB and UVC radiation. Once you get shorter wavelengths than that, the cornea just doesn't let them in. Those are really damaging radiations. And that's why this process takes years and years to develop, because the cornea is only letting through about 2% of these damaging UV radiation when you're appropriately exposed. So that's why it takes a really long time for this process to develop. I wish there was such a thing as a Google Glass that measured your UV radiation and we could just have people wear them for really long periods of time, but they don't exist. But anyway, what does exist is studies such as this by Zenkel. And what they did was they took cells from the ocular surface and they exposed them to UV radiation, various doses, and they found that UV radiation upregulates things like LOXO1, elastin, fibrillin. All these things down here are large molecules that are typically found in exfoliation material. And lo and behold, UV radiation causes an uptick in all of those molecules. So, but just to shift gears here a little bit, for every case of exfoliation glaucoma in the United States, there's roughly 5 cases of primary open-angle glaucoma. And if we were to make a natural history diagram for primary open-angle glaucoma, it's more challenging because we don't really have a biomarker like we do for exfoliation glaucoma. I think this is why Dr. Rich's strategy of first focusing on exfoliation syndrome was really wise. But for primary open-angle glaucoma, the disease is more common. The questions are more difficult because the only biomarker we have is that the optic nerve gets more deteriorated, and that happens across the spectrum of intraocular pressure. Certainly, blindness happens, and it is one of the leading primary open-angle glaucomas, one of the leading causes of blindness. But for primary open-angle glaucoma, the burning questions are different. The questions are, why does the pressure go up even though there's no obvious physical obstruction of the drainage angle? Why do people develop progressive optic nerve degeneration even when the intraocular pressure seems to be normal? And could we possibly protect the optic nerve regardless of what the prevailing intraocular pressure level is? So in conclusion, because I think time frame for these kinds of talks can't go on forever. But I would say this: the Glaucoma Foundation supports research that provides insights into how exfoliation material is formed in exfoliation glaucoma. And we have received a grant from India looking at this UV radiation hypothesis in more detail. The foundation wants to support researchers who are developing models of exfoliation syndrome because, as Sal said, we don't really have a good UV Geiger counter for the eye. So we hope that we can develop models for that exfoliation material can form either in vitro or in an animal model. And perhaps it might involve UV radiation. Perhaps it might involve other things that other researchers are thinking about, but with the idea that they can develop the exfoliation material outside of humans. Exfoliation syndrome, though, is different compared to primary open-angle glaucoma. And primary open-angle glaucoma is an even bigger problem in the United States and poses unique sets of challenges. So now the foundation will branch out and support researchers who want to develop strategies to understand primary open-angle glaucoma, understand why the optic nerve deteriorates regardless of what the intraocular pressure is, and how to either prevent that or develop strategies to restore optic nerve function in those patients. So I'll stop there. Thank you for your attention. I'll be happy to take any questions. Maybe I'll stop sharing my screen. There we go. Great.
This was quite brilliant, Lou, and I know that everybody will share will join me in thanking you, because it was a great presentation, beautifully done. And we have still 10 more minutes before our end time. So if anybody has any other questions to Lou, just unmute yourself and ask a question. Yes, Ken.
Unmute, Ken.
I unmuted him.
Great.
Sure. Hi, that was just really wonderful. It was so well explained to a layperson, and I'm ready to send Elena some more money now. That was great.
Thank you. That's great. That's great. Paul. Unmute Paul.
I'm muting Paul.
Wait 1 second, Paul.
I can't unmute Paul. Okay, now he's
Paul
unmuted.
unmuted himself.
Okay. So that was a great talk, and of course, I know you have a lot more to say about geography and sunlight and all that. But a couple of interesting points, I think. We've had done over the course of a lot of years. We've looked at a lot of non-human primates, and we've looked at them, ones that are kept in captivity indoors all year round for decades, and ones that live outside all year round for decades and live in jungle environments where they're out in the sun at least part of the time. And we've never seen exfoliation in a non-human primate. And it's very, very rare that you see elevated pressure, or what we would call primary open angle glaucoma, in a non-human primate. Yet in so many other ways, they're so much like us, and their eyes are so much like our eyes. And so you're wondering, what is the difference? I mean, humans and monkeys are not the same, obviously, but if you could figure out why they don't get it and why we do, and they can live a long time. They live to be old, decrepit monkeys. And yet you don't see the disease. And I know there's a group in Florida that claims that they have a strain of monkeys that develop high pressure and glaucoma, but not a lot of people have seen those animals, so we don't really know what we're dealing with.
So thank you for the question, Paul. And two things. First, remember that I showed the map of Africa, and then in Tanzania, there was zero exfoliation. And so one has to wonder, monkeys are tree-dwelling mammals for the most part. Maybe they're being naturally, they have a natural pair of sunglasses, and that there's not a lot of reflection in the environment that they're in into the eye. But I can't prove that. But number two, I am working with a group from southern China that has a colony of monkeys, and they're macaques that are very similar to the macaques that were described in southern Florida. And I was supposed to give a talk at Arvo on this, and with COVID, it got squashed. But you're quite right. Those monkeys seem to get primary open angle glaucoma. In fact, they get all kinds of human diseases, not just glaucoma. They get macular degeneration, they get diabetes, they get diabetic retinopathy, they get macular degeneration, and we don't know why. So I too have not seen exfoliation in those monkeys from southern China. But again, it might be because of the lack of ocular UV exposure, and that's the reason why.
Okay, thank you.
Hey Lou, it's Dave Fellows here.
Hi, Dave.
Question, is there any difference in the diurnal fluctuations between patients with exfoliation and just primary open angle, or is it not studied?
So you mean diurnal variation in intraocular pressure?
Yes.
I don't know that literature cold. Perhaps Paul knows it better than me, but my gut impression is that the diurnal variation in eye pressure is higher in exfoliation than it is in primary open angle glaucoma. And this is largely because just turning the room lights on and off, if you've got exfoliation material sitting on your lens, the iris rubs up against the lens, and it's like sandpaper rubbing up against a rough surface. It just liberates pigment and exfoliation material, and that clogs the drain, and the pressure can go up very high on fairly short notice in exfoliation syndrome.
Well, also, if your resistance is higher, then just by having a higher resistance, the fluctuation in response to the fluctuation that we all have with rate of aqueous formation at time of day is going to be magnified. So there are biological aspects to it, but there's also just a physics aspect to it. And that's just the nature of the way pressure and flow work.
Thank you.
Anybody else? Let's do Pat first, and then we'll do Reza. Go ahead, Pat. Oh, you muted. Let me unmute yourself, Pat.
Am I unmuted?
Now you're unmuted. Yes.
Okay. I want to thank you. Being a total layman, it was very, very interesting. And I feel like I have a concept of the words and the issues. So thank you very much. And my eyes didn't glaze over. I just got very nervous with what's going on in my head now, but that's a separate issue.
Thank you, Pat. And now Reza.
Yeah, thank you. It was a wonderful, wonderful talk. Just two questions. One is there any evidence of the exfoliative material in other parts of the body on those who've had the UV exposure? And I guess, well, maybe not relatedly, but any clinical advances in terms of treatment, notably with outflow drops and so forth, to get the exfoliative material out of there to open up the drainage?
So great questions, Reza. The exfoliation material is found in structures outside the eye. You can find it in the heart, you can find it in the lungs, you can find it in skin, but it takes an electron microscope to find that material. And that, I believe, is because the motif of the way the body works is different there than it is inside the eye. Go back to the topsoil analogy. You have a surface cells, and then you have the underlying topsoil, and then there's vital structures underneath that. And so the basement membrane doesn't face fluid-filled cavities in the lungs or in the heart or in the skin. And so it I think that physically confines the ability for the material to accumulate in those structures. And that's why you don't see a lot of it there. What was the second question again?
It was just with regard to any clinical advances, notably with regard to the outflow drops.
So we would love to be able to disassemble this stuff. Dr. Rich did this amazing study. I mean, the guy is brilliant. He took the material and he exposed it to like 99% formic acid, and the stuff doesn't dissolve. It's like that movie Super Size Me, when you take the McDonald's French fries and you put it in a jar. They stay looking like French fries for like 90 days. Whereas if you get French fries from the corner deli, they turn to mush in a day. So there's something about this material that makes it really hard to get rid of. Now, what people have done is they've taken like a surgical vacuum cleaner and gone into the eye and tried to suck this stuff out. And it works for about 6 months or so, but because you don't stop the material from accumulating, it reaccumulates and the pressure goes back up. So it is going to be challenging to find something that could disassemble the stuff. And I've talked to Dr. Rich about this, and he's like, someone will figure it out. And you never discount what Dr. Rich says, because that's what the foundation does, is we try to find people coming out of left field that might be able to figure this kind of stuff out. But that's what we need. And we've even talked about supporting studies of early lens extraction in patients. In other words, take out the lens even before the cataract starts to form in these eyes that have early exfoliation, because if you just sort of debulk the lens, then you reduce the amount of redistribution of exfoliation material. You give the drain a chance to recover, and maybe those patients might do better. Then when we start talking about the nuts and bolts of a study like that, it gets very complicated very fast. And there's a lot of feasibility issues. But we have during some of our think tanks, spent many minutes, and maybe an hour, Paul, talking about how to design a study like that. So yeah, we think about those things all the time.
Thank you. That's great.
Well, I see your raised hand. You have a question?
Well, I just wanted to thank Lou for the presentation. It was magnificent. You know, it's always thought-provoking and makes you think. And you know, when you focus on it, it's great. I'm grateful for being able to say, well, we've done this study as well, and yes, they're pursuing that, because I think it gives the donor community confidence that, you know, Lou can't tell us everything that's happening. So, but do understand beyond what he's presented. They're worrying about, you know, lush, you know, you never thought you'd talk about the lush green fauna and flora, you know, in Africa and the equator, and, you know, but it all comes into play. And if we gave him 5 hours, he still wouldn't be able to cover everything. But it's really, to me, inspiring, and, you know, really gives me a sense of confidence that the money we're pouring in to address this issue, it'll never be enough until we're done. Because what rate limits the cure is how much money we push into the envelope to have all the questions answered. He said it's a difficult study. What he means is it's an expensive study. and sometimes that's really the issue. So money can solve a lot of problems and at least push it forward. And, you know, look at this vaccine for, you know, COVID-19. You know, in less than a year-ish, there's going to be a couple of successful kind of candidates. That was a function of focus like a moonshot and money. It wasn't just smarter people worked on this than who worked on AIDS. That took longer. I think that's probably the case and this comment is mostly to thank Lou and to put the philanthropic community also with the thanks, because that's the fuel that makes the car go.
That's exactly right.
Thank you, Sal, for your kind words. Mission accomplished. And another great analogy is AIDS. So, right, what you're saying is right. Once you put critical mass behind an idea, you can solve almost any problem. So, no, we haven't cured AIDS, but guess what? People are not dying from it anymore. We have this triple antiviral therapy that keeps it at bay. Every once in a while, we read in the newspaper about another patient who's virus-free. Maybe we'll get there someday, but it was only because of advocacy and people banging on the doors of Congress saying, let's put money behind this. We've got to stop this problem. And I think that's the way COVID will end up too. Maybe it won't be a vaccine. Maybe it will be an antiviral, but it will be something. We will beat this down because we've got to put the money behind it. And I thank the board for putting money behind glaucoma and for helping us to answer critical questions there too, because we do have a lot of patients suffering from this, and we need to continue to fight the fight against it.
Beautifully said. And of course, I want to echo what Lou said. Of course, I want to thank Lou so much for this absolutely brilliant presentation, and echo what Lou said. Yes, we need more money. So think about it, what you do, and add at least 10% to what you do, so we can continue solving this problem. And thank you all very much. It's our time. Thank you, Lou, again. Brilliant, brilliant presentation. And thank everybody for joining.
Okay.
Thank you, Elena.
Thanks for organizing, everybody.
Thank you. Have a great day, everybody.
Okay, take care. Bye-bye.
Bye-bye.